Hello everyone! Today, we're diving into a fundamental concept in chromatography, void volume. It sounds a bit technical, but understanding it is key to interpreting your results. Let's break down what void volume is and why it matters in your chromatographic analyses. So, what exactly is void volume? Imagine your chromatography column. It's packed with a stationary phase, like tiny beads. Void volume is simply the space around these beads. It's the volume of the mobile phase that is not interacting with the stationary phase. Think of it as the empty space within the column that the solvent and samples can flow through without resistance or interaction. We often call this the extraparticulate volume or interstitial volume. It includes the volume of the tubing leading to the column, the injector, and the detector cells as well. This total non-interacting volume is crucial. Why is it important? First, the void volume tells you when the very first compound that doesn't interact with your stationary phase will come out of the column. This compound is called an unretained compound. It travels at the same speed as the mobile phase itself. Second, void volume is essential for calculating the retention factor, K, of your analytes. The retention factor measures how much longer a compound stays in the stationary phase compared to the mobile phase. Without knowing the void volume, this calculation is impossible. Third, it helps you assess the efficiency of your column. A well-packed column will have a consistent void volume. Fourth, changes in void volume can indicate problems with your column packing or system, acting as a valuable troubleshooting tool. How do we determine void volume? You inject a compound that you know for sure will not interact with your stationary phase. This compound is called a void marker or unretained marker. Common void markers include deuterium oxide, D2O, in reversed phase HPLC, or a very large molecule like blue dextrin in size exclusion chromatography. For UV detection, a small, highly polar, non-retained compound like thiourea is often used. The time it takes for this unretained marker to elute from the column is called the void time, T0 or Tm. Multiplying this void time by the flow rate gives you the void volume, Vm. This represents the minimum time any compound can spend in the column. Any compound that elutes before the void time is considered to be excluded from the column, which is common in size exclusion chromatography. Understanding this baseline elution time is fundamental to understanding all other retention times in your chromatogram. It provides the reference point against which all other compounds' retention is measured. So there you have it, void volume demystified. It's a simple concept with big implications for accurate chromatography. If you found this explanation helpful, please consider giving this video a like and subscribing to the channel for more insights into analytical chemistry. Your support helps us create more content like this. Thanks for watching.